When you read a nutrition label on the back of a package, there's this expectation of an exact, honest accounting.
Right, like a receipt.
Exactly. It feels like a bank statement. You look at the calories, it says 0, and you just think, perfect. It is a clean transaction.
Yeah, 0 calories deposited, 0 impact on the balance sheet of your body.
Right. And I mean, 0 means 0. We desperately want the math to be that simple. But when you step into the world of neurology and cellular biology, well, that simple bank statement becomes completely useless.
Oh, completely. You are looking at a metabolic landscape that is incredibly complex and, honestly, a bit chaotic.
And that biological chaos is exactly what we are unraveling in today's deep dive into the source material. We are examining 2 brand new, frankly groundbreaking studies. One is out of Brazil, published in the medical journal Neurology, and there is a parallel study from the University of Cambridge.
And together, they're pretty much challenging everything we thought we knew about artificial sweeteners.
Yeah, they really are. Okay, let's unpack this. Because there is a massive paradox sitting right in front of us. Millions of people, and you listening right now might be one of them, consume low and no calorie sweeteners every single day.
And we do it specifically to protect our health.
Exactly. To lose weight, to avoid sugar crashes, to just make a supposedly better choice. But these new findings suggest we might be paying a hidden neurological cost.
Which is a terrifying thought.
Right. So I want you to imagine your morning routine for a second. Maybe you grab a diet soda for a caffeine hit, or you have a light artificially sweetened yogurt with your breakfast. If these sweeteners are marketed as 0 impact, how are they suddenly showing up on a neurologist's radar?
Well, what's fascinating here is that the scientific community is finally evolving its perspective on what it actually means to test a food additive.
Meaning they used to just look at weight.
Yeah, for decades, the entire focus of artificial sweetener research was centered almost exclusively on weight loss and acute toxicology. The only questions being asked were basically, does this make you gain weight? And will a massive dose of this poison you immediately?
Right, like will you drop dead from eating it?
Exactly. But now scientists are moving far beyond that simplistic view. They're measuring the long-term systemic impact of these chemicals on complex biological ecosystems, like the brain and the gut microbiome.
So we are finally asking what happens when a human being processes these synthetic compounds day after day, year after year.
Precisely. And to answer how they get on a neurologist's radar, we first have to look at the staggering numbers from this new Brazilian study.
Yeah, let's get into those numbers. Because this wasn't some small, 6-week trial with 20 college students. Researchers in Brazil followed more than 12,700 adults.
With an average age of 52, which is a really critical demographic for this kind of research.
Right, and they tracked their dietary habits and cognitive function over an 8-year period.
That 8-year tracking period is what makes this so robust. I mean, it gives you a wide enough window to observe subtle cognitive changes over time that shorter studies just completely miss.
So they tracked the consumption of 7 very common low and no calorie sweeteners. We're talking aspartame, saccharin, sulfame K, erythritol, xylitol, sorbitol, and tagatose.
Basically, the exact ingredients you will find in diet drinks, flavored waters, yogurt, and ultra-processed foods.
Yeah, breakfast cereals, packaged snacks, all of it. So the researchers divided these 12,000 people based on their consumption levels. The people who consumed the least took in about 20 milligrams a day.
Which is very little.
Right, but the people who consumed the most averaged 191 milligrams a day. Wow. And the finding here is a bombshell.
It really is.
The high intake group, that 191 milligram group, saw their memory and mental sharpness deteriorate 62% faster than the low intake group.
A 62% faster rate of cognitive decline.
Yeah. The research team equated this to roughly 1.6 years of additional brain aging.
Which is a highly significant acceleration. We are talking about a measurable degradation in how the brain ages, specifically in executive function, memory, and just general mental sharpness.
Well, wait, I have to push back here. Because I am looking at this 191 milligram number, and I know our listeners are doing the math in their heads right now.
It's not as high as it sounds, is it?
No. 191 milligrams is roughly the amount of sweetener found in just one single can of daily diet soda.
Just one.
Wait, so we aren't talking about drinking a 12-pack a day. Just one can's worth is linked to aging your brain an extra year and a half. Wouldn't we have an epidemic of early-onset dementia by now? I mean, we've been drinking aspartame since the 80s.
That is the exact distinction we need to make here. We have to separate clinical dementia from accelerated subclinical cognitive decline.
Okay, what does that mean in plain English?
The study is not saying one diet soda gives you Alzheimer's. It is measuring the depletion of your cognitive reserve. When we say aging 1.6 years faster, we are talking about subtle deficits.
Like forgetting where you put your keys.
Yeah, or it's the difference between quickly recalling a name versus struggling to find the word for a few seconds. It's a slight decrease in your ability to multitask or solve complex problems under pressure.
Oh, I see.
Over a population of millions, that 62% acceleration shifts the entire baseline of human sharpness.
That makes a lot of sense. So as the study's author, Dr. Claudia Kimisumoto, noted, these things are seen as healthy sugar alternatives, but they may have profound negative effects on the brain's health over time.
Exactly.
So knowing this startling top-line number, the natural next step is figure out who is most at risk. And also, do all sweeteners act the exact same way?
Right, because the data revealed some very specific vulnerabilities.
Yeah, the link to cognitive decline was surprisingly strongest among adults younger than 60 and among people who have diabetes.
Which is tragically ironic.
It is. Diabetics are the primary target demographic for the marketing of these sugar substitutes. They are told this is their safe option.
Absolutely. It's marketed as the responsible choice for them.
But out of the 7 sugar substitutes tracked, 6 were associated with this faster cognitive decline. But here's where it gets really interesting. There was one lone exception that did not follow the pattern.
Right, tagatose.
Yeah, tagatose. If 6 of these chemicals act like a fast-forward button for memory loss, but tagatose doesn't that imply it's the specific chemical structure, not just the abstract idea of sweetness, that's messing with our biology?
This raises an important question about how our bodies process different chemical structures. It absolutely points to the shape of the molecule.
Oh, so.
Think of your cellular receptors and metabolic enzymes like highly specific locks, and the molecules you ingest are the Ps.
Okay, locks and keys.
Aspartame, saccharin, and sucralose have molecular shapes that trigger very specific biological pathways when they enter the human digestive tract.
The tagatose is different.
Yeah, tagatose happens to have a different stereochemistry, a different physical three-dimensional shape. Its particular key simply does not fit into the same biological locks that trigger the negative downstream effects we're seeing with the others.
Oh, wow. But I feel like before we crown tagatose as the new miracle sweetener, we have to look closely at a crucial caveat from the Brazilian researchers.
Yes, the causation versus association caveat.
Right. This study shows a powerful association, not direct causation, meaning it proves that people who consume more of these 6 sweeteners experience faster brain aging, but it does not definitively prove that the sweetener molecule is directly traveling to the brain and attacking neurons.
Precisely. There are always confounding variables in observational studies.
Like lifestyle factors.
Exactly. Perhaps people who consume a diet soda every day are also getting less sleep, or they are experiencing higher chronic stress, or eating more ultra-processed foods in general.
So the researchers are clear that the pattern of cognitive decline is undeniable, but we just don't know the physical mechanism yet.
Right. A statistical link is not a biological mechanism. We still need to know how this damage is actually occurring inside the body.
Well, if the Brazilian study shows us the smoke, we need to find the fire. If the sweetener isn't crossing the blood-brain barrier and directly attacking brain cells, the damage must be starting somewhere else.
And where is the body's primary chemical processing plant?
The gut. Which brings us perfectly to the University of Cambridge study. This was released right alongside the Brazilian findings, and it looked at exactly how these chemicals interact with our digestive system.
The Cambridge study provides the missing piece of the puzzle. It shifts our focus from the brain to the gut microbiome.
Which is like this delicate garden of vast microorganisms, right?
Yeah, it's the vast ecosystem of bacteria, fungi, and microorganisms living in our digestive tract. And what modern science is increasingly proving is that the gut and the brain are deeply chemically connected through something called the gut-brain axis.
So the Cambridge researchers ran lab tests where they exposed 25 different species of human gut bacteria to 39 different artificial sweeteners.
And the results were intense.
They really were. About 75% of the sweeteners altered at least one species of Listeria.
Crucially, they were actively slowing down or completely stopping the growth of specific microbes associated with digestive health and blood sugar regulation.
Okay, let's use a metaphor here. If we've been told these sweeteners just pass right through us like water, how is it that they are acting like a weed killer on the bacteria meant to regulate our blood sugar? Like, is swallowing an artificial sweetener actually like dumping a bucket of acid on those bacteria?
No, it is far more insidious than that. To borrow an assembly line metaphor, think of your gut bacteria like workers on a factory assembly line.
Okay, and their job is to sort nutrients.
Right, to sort nutrients and regulate your metabolism. The sweetener isn't acid. It's more like dumping thousands of fake plastic bolts onto the conveyor belt.
Oh, man.
The factory workers, the bacteria, pick up these plastic bolts thinking they are real nutrients. They try to process them, but they obviously can't.
So the fake bolts jam the machinery.
Exactly. The bacteria waste all their energy trying to metabolize a synthetic chemical, which effectively starves them or shuts down their replication cycle. They can no longer do their actual job of regulating your blood sugar.
That perfectly explains Dr. Sanja Blash's quote from the Cambridge study. She said, "Sweeteners are often marketed as metabolically neutral, but our study challenges this idea."
Because we've been told for decades that if a chemical doesn't contain human calories, it's harmless.
Right. Meaning our human cells can't extract energy from it, so it just passes through us like a tiny glass marble.
But we completely forgot about the bacteria. Our human cells might not digest these chemicals, but the millions of bacteria in our gut absolutely interact with them.
So a substance lacking calories does not mean it lacks biological activity.
Exactly. It triggers profound reactions in our gut flora.
Okay, this is where we need to connect the two studies for everyone listening. We have the Brazilian study showing a 62% faster cognitive decline, and the Cambridge study showing jammed up gut bacteria. How do jammed up gut bacteria cause the brain to age a year and a half faster? What is the actual chemical bridge between the stomach and the brain?
It comes down to neuroinflammation and the specific byproducts that gut bacteria produce.
Okay, break that down for us.
Healthy gut bacteria consume dietary fiber and produce compounds called short-chain fatty acids, or SCFAs.
And these are good for us.
They are incredibly protective. These SCFAs travel through the bloodstream, they cross the blood-brain barrier, and they essentially tell the brain's immune cells to stay calm.
So they reduce neuroinflammation.
Yes. But when you introduce artificial sweeteners that jam the machinery and kill off these specific healthy bacteria, your SCFA production drops.
I see. So without those fatty acids acting as a sort of chemical fire blanket, the brain's baseline level of inflammation rises.
You got it. Chronic, low-grade neuroinflammation is one of the primary drivers of accelerated brain aging and cognitive decline.
That is wild. The sweetener doesn't even have to touch the brain to damage it. It just has to kill the gut bacteria that act as the brain's protectors.
It is a terrifying domino effect.
And the Cambridge study didn't just stop at sweeteners in isolation, right? Because nobody consumes these chemicals in a sterile vacuum.
No, we drink them alongside our morning coffee, our breakfast, or daily prescriptions.
Right, which leads to what I think is the most complex and surprising finding of this entire deep dive.
They documented the multiplier effect. They essentially mapped the reality of the modern human diet.
The researchers identified over 100 instances where caffeine, everyday flavorings, and common medications actually amplified or reduced the sweetener's effects on our gut bacteria.
The interactions are incredibly complex.
Yeah. They found one incredibly strong interaction involving a low-calorie sweetener called isosteviol.
Which is used heavily by the food and beverage industry.
Right. When isosteviol was combined with duloxetine, which is a very common antidepressant medication, it significantly suppressed two highly beneficial gut microbes.
Microbes that are vital for digestive health, blood sugar regulation, and immune function.
So what does this all mean? How on earth do an antidepressant and a flavored water additive team up to attack my immune system?
If we connect this to the bigger picture, it comes down to synergistic toxicity.
Synergistic toxicity.
Yeah. When a microbe is exposed to a single foreign compound, like the sweetener alone, it has defense mechanisms. It uses cellular pumps, called efflux pumps, to push the unfamiliar chemical back out.
Okay, so it spits it out.
Right. But when you introduce a second complex molecule simultaneously, like the duloxetine, you overwhelm the microbe's defenses.
The pumps get jammed.
Exactly. The antidepressant might occupy or block the microbe's efflux pumps, which then traps the sweetener inside the bacteria. The combination creates a toxic buildup that neither chemical would achieve on its own.
Wow, it's like we are inadvertently running a complex chemistry experiment in our stomachs every single morning.
We really are.
If someone takes their prescribed antidepressant with a diet-flavored water, they have absolutely no idea they might be actively suppressing their own immune function.
None at all.
I mean, the label on the water just says 0 calories. It doesn't say, warning, reacts with antidepressants to paralyze your gut bacteria.
Professor Kiran Patil, the senior author from Cambridge, summarized this perfectly. He said, "Our study suggests that artificial sweeteners don't just pass through the body passively. They can interact with gut microbes, and these effects can be amplified or altered by other substances like medications."
Which really validates why both sets of researchers are emphasizing how much remains unknown and why more research is needed.
It's no longer just about testing one isolated ingredient in a lab. We have to study the entire ecosystem of modern diets to understand how these combinations create unexpected cascading physiological effects.
And we should be clear, both the Brazilian and Cambridge researchers are calling for massive additional research before we establish undeniable causal links.
Right, they aren't saying the sky is falling and we need to ban every diet drink tomorrow.
No, they are being meticulously measured, as scientists should be, but they are pointing to a rapidly growing body of evidence. We have to seriously examine whether these products, which are heavily marketed as the responsible, healthy alternative to sugar, are actually driving a slow-motion public health crisis.
Through completely unintended biological consequences.
Exactly. So let's synthesize everything we've uncovered for you listening today. The foundational takeaway from this deep dive is that the marketing of low and no-calorie sweeteners as a free pass for your health is structurally collapsing under the weight of new science.
Absolutely collapsing.
We started with the Brazilian studies showing that the equivalent of just one single daily diet soda is associated with 1.6 years of accelerated brain aging and a 62% faster deterioration of memory.
And then we provided the biological mechanism for that cognitive decline via the Cambridge study.
Right, the gut connection.
Yeah, these supposedly neutral chemicals are acting as synthetic decoys in our gut microbiome. They jam the metabolic machinery of the bacteria responsible for blood sugar regulation and immune function,
stopping the production of those protective fatty acids that keep brain inflammation in check.
And worse, they interact dangerously with everyday medications to multiply that damage.
So the bottom line is that these substances are active, disruptive participants in our biology, not passive passengers. Which brings me to a final, really provocative thought I want to leave you with today.
Okay, let's hear it.
Something to really mull over the next time you are standing in the grocery aisle. For over a century, modern medicine has defined a calorie simply as a unit of heat.
Right.
A unit of raw energy that a human cell can burn.
It is a strictly thermodynamic measurement, yes.
But if a zero-calorie chemical enters your stomach, permanently alters the bacteria responsible for processing other nutrients, paralyzes your immune response when combined with medication, and indirectly accelerates the aging of your brain by stoking inflammation.
Could our medical definition of a calorie be entirely fundamentally flawed?
Yes. If a zero-calorie chemical changes how your gut bacteria processes nutrients, it forces us to ask if measuring heat energy is the right metric for human nutrition at all.
It's a huge paradigm shift.
If a label says 0 calories, but the chemical inside fundamentally changes how your body extracts energy and manages cellular health, then looking at the nutrition label on a diet drink isn't just incomplete information.
No, it might be telling us the exact opposite of what the biological ledger is actually recording.
Think back to that bank statement we talked about at the very beginning. If the statement says 0, but the bank itself is quietly devaluing your currency and dismantling the vault behind the scenes, well, that 0 is a dangerous illusion.
It is a fake number. Biology isn't a bank. It is an intricate, highly reactive ecosystem. And we are honestly only just now learning how to read the actual ledger.
But we really are. I hope this deep dive into the source material encourages you to look at your pantry in a whole new light. The label might promise you 0, but your biology is definitely keeping score.
Always keeping score.
Thank you so much for joining us. Keep questioning what you read, and we will catch you next time.